So BLAST. The first thing that you have to keep in mind is that BLAST is not Google. That is the most important thing that you have to keep in mind. BLAST is not Google. And it's not my phrase, I took it from somewhere else, but the basic point is in Google, you write something, and in general, the first hit, the first or second are what you are looking for. BLAST is not Google. BLAST has many options, and the rest will depend on the option, and you have to choose the option carefully. Basically, there are two ways to use BLAST. One is going to the website of NCBI. Well, there are more than one website. There's probably one in the European database, but using something online or using in your computer. Let's say the two options are you use someone else's computer via web, or you run in your computer. The trick is in using on your computer is that you can have your own databases, and you can you have your own computer. You don't have to share the computer with other people. The public websites have many people using them at the same time, so you don't control how low they have, how much other people are competing. And even there are some options that you can use locally that you don't have in other cases. You don't have what I mean. You have other options that you don't have in the web version. The command line version is not something that we're going to see here because it's kind of complicated, but let's see, depending on the time. So for now, we are assuming that you are going to use the web version, which by the way is this one. This one. Okay, we are going to look at this. By the way, there is something here. Download BLAST you can get here. Let's see. And you can use from your application. You can have it in the cloud and all the new fancy things. So you can download, and there are new BLAST that we will explore later. Do you have difficulty running high volume BLAST? For example, you want to look for 1 million, even 10,000 reads, 10,000 sequence is hard to put in the server. Do you have data that is proprietary? Do you have your own server? Do you have security of intellectual property? Well, that was in my case, since I was in my previous life, I was working with some company data. We had to do it locally. Okay, but here you can see, and I don't know if there is a version for Windows. Let's see. Okay, let's go back. So, in fact, I will be moving between these two slides. So, depending on the alphabet of the sequence, we have, let's say, 2 or 3 options. The basic ones are BLAST nucleotide and BLAST protein. Here, nucleotide BLAST, protein BLAST. And then we have something in between that I wanted to take a minute to say. So, BLAST, okay. Let's say BLASTX. Let's start with the basic thing, and then we go to the BLASTX, that's the translated one. That here, no? BLASTN, nucleotide, BLASTP, protein database, BLASTX, search nucleotide in protein databases. Each query is translated. So let me show you what I mean with this, because I have waiting for. In this Artemis here, I hope you can see it. Can you see it?
Yes.
Okay, great. So let's make it a little bigger. In this part, I have the gene, but maybe this second part here is more useful. If I move the scroll, I think I can make a zoom here. Yeah. So, okay, here we're more or less at the same level. In the center, you have the nucleotide sequence. Okay, this is the sequence that I loaded from the file mouse gene. I can give you this. I can give you later this. In the lower part, I have the reverse complementary. So this is A becomes T, T becomes A, so of the two DNA strands, we only store one because we can calculate the other automatically. Okay, but there are two DNA strands all time. We write one, but we know that there is another. Moreover, on each of these sequences, we can take 3 letters, and that will give us an amino acid, no, codon. But if I take the second, third, and fourth, I will have another amino acid. If I take the third, fourth, and 5, I have another amino acid. Okay, so in other words, the same sequence can be transformed to amino acid in 3 ways. 1, 2, 3. So there are 3 reading frames in the forward strand and same in the reverse strand, except that in this case they go from right to left. Okay. So if you know the nucleotide, it is very easy to predict what will be the protein. Okay. There is a code for that, a genetic code. But if you have the protein, getting to the nucleotide is not easy. Okay. Because one amino acid can have more than one way to encode, and the number of combinations grows very quickly, so it is a hard problem. So from nucleotide to amino acid is easy, from amino acid to nucleotide is hard. Then for every DNA, you have 6 reading frames. So the BLAST-X, what they do, you take a nucleotide, you give a nucleotide, you have the sequence, and then the BLAST system translate automatically that sequence into 6 theoretical proteins, 6 virtual proteins, if you like. The one that we saw here. This one can be this, or okay. In fact, we will search for this gene, for this nucleotide, and we will see what we find. Okay. So that is the idea of BLAST-X. Then there are a couple of more tricky. Searching protein in nucleotide database, then it is super complicated because essentially they have to take the protein that you are, no, they have to take the nucleotides in the nucleotide database, translate the nucleotide into protein in the database, that are millions, and then search your sequence to everything. For every protein, for every nucleotide in the database, you get 6 possible proteins, and you compare your protein to these 6 possible proteins. And the worst is this one. You have nucleotides, and the database is nucleotide, but you translate each query and each subject into proteins, and then you compare everything. You compare nucleotide to nucleotide, but assuming that they are coding for proteins. Tamam?
Maybe.
Maybe what?
Not tamam. Can you go to Artemis,
Yes, sir.
please?
That I like when people ask questions.
Okay. We assume that ATG may produce amino acid, right? The first.
Yeah.
Okay.
Yeah.
What if TGG not produce an amino acid?
No, every 3 letters will produce unless it's a stop codon. But there is still a correspondence between the 3 letters and a stop codon. It's a symbol. There is at the symbol level, you are translating a sequence from DNA alphabet to sequence of protein, that the alphabet from nucleotide to amino acid. Okay. So except for the stop codon like this one or this one, all the translation correspond to an amino acid. If you think that this is not, this is a stop codon, yeah, fine, but this is still a symbol that we can handle. Because when you compare proteins, maybe, or here, one more. When you compare two proteins, yeah, maybe the stop codon is annoying, but this can be a mismatch, and the rest of the protein is fine. Why this will be relevant? Okay, first, I don't know if I explained myself better this time. My first point is that given a DNA without any concept, let's say that you have a PCR product and you don't know what is that. That PCR product without a start or stop codon, if you don't have any identity, any idea what is inside, that fragment of DNA can be part of a protein in 6 different ways, and you don't know a priori which one is the correct one. You follow me? Sometimes in this translation, you will get stop codons. Well, that can happen either because, for example, you get the end of a protein and the beginning of the next one, or there is something else that happened that is called a pseudogene. You know what is a pseudogene? Essentially, how I see it is that you have a DNA sequence that was a protein, but then some insertion happened and you got a stop and the protein following a different, if you insert a letter here, what happens? If you get a single letter insertion, you will have that the protein that was in one reading frame will jump to another reading frame. And you will probably have a stop in between. Well, not necessarily. But the point is that you have a stop codon is not a reason for not looking in the database.
Okay, my bad. It was a stupid question. And I have another stupid question.
Ask stupid question. This is the lemma of this class. Ask stupid questions.
Why we shift it just 3 nucleotide? For example, can't we shift more? Right? I mean, we produce 3 amino acid sequences.
Yeah, because the codon size is 3. Since the codon size is 3,
but
if you shift 1, you get a different reading frame. If you shift 2, you get another reading frame. But if you shift 3, you get back to the first one. Let's say you can start in the first letter. This is the first letter? No, let's start in the first. You can start at the first letter and count 3. You can start in the second letter and you get the second reading frame. You can start in the third letter and you get another in the third reading frame. But if you start in the fourth letter, you are back in the first reading frame. And that is basically because the codon size is 3. So after you finish the 3, when you get to the fourth, you are at the start of
so we start to repeat ourselves after
exactly.
Okay, exactly, I get it now.
Exactly. Yeah.
But is it repeating? Like we were at
not repeating, but you're back again in the same reading frame. Yeah, not technically repeating, but
okay. First three sequences contain all of the possibilities, right?
Yeah, the first 3. I mean, not the first 3 sequences. The 3 reading frames are all that can be in the forward strand. Remember that there are other 3 in the reverse direction, no? You have 3 in the forward and 3 in the reverse. Yeah, no?
What's the question?
That given a DNA, we can translate to protein in 3 ways in the forward strand, way 1, 2, and 3. But since DNA double strand, there is another strand going in the reverse direction that can also encode for protein in the opposite direction. In other words, for a given DNA, for a given fragment of DNA, you can have 6 possible proteins.
Okay.
By the way, viruses do that a lot. One thing that you find in viruses is that when the same sequence of DNA or RNA, they encode for more than one protein. And that is super hard to do, but it's kind of, imagine that you are writing a text in I don't know, in English, but at the same time, you are using the English word to write something in Turkish. Like part of your text in English will be something in Turkish. It's kind of very crazy. In general, you don't have the case that two proteins touch each other, or if they touch very little. In general, you don't have a protein inside a protein, except in virus. In virus, that tend to happen a lot. It is important that you ask all these questions now, because, well, mainly because there's something that I've been doing for so many years that to me are super obvious, but they're not obvious. I mean, I don't see that part that are not clear. I don't see. So it is super important that you ask questions, and we profit that we are a few people here, and it is time to ask questions. Well, that was essentially to explain what are these things here in between. But don't worry, we are going mostly to use either nucleotide or protein. Let's start with this one. Maybe I can make it a little bigger. This COVID message, of course, is only in the last year. They put it in every page, but let's see what do we have here. Okay, you see here, there are some options that you can see there, all kind of help that I will ask you to read later. And no, the other way. First thing, if you choose the wrong one, don't worry, you can choose BLAST-P. All the options are here. Okay, all the variants of BLAST that I show you are here. Then you can put the sequence and in different ways. How can we do?
Is it like all sequence?
Yeah, you can put the accession, or you can put the FASTA sequence. GI, remember, is the old the gene bank identifier is the old way to do it. So you can put the sequence here, and since it's FASTA, you can put more than one, and you can put, you say numbers. You can put more than one, and they will search all of them. Let me see. I have somewhere here. Okay, to make it easy, I will just take the sequence that I just showed you, the mouse sequence. I have others, but in fact I have one later. But just to give you an idea, I am taking part of the genome that I just show you in Artemis here. Okay, I cut and paste the sequence. Because I am assuming, for example, that this is a sequence that I got from an experiment. I isolated some sample and I sent to sequencing, and I want to know what they have. Now, to be honest, this is probably going to be done by you. Most of the companies that sequence, they will do the blast automatically for you. They don't even ask. You sequence something, and the company will send you the sequence, and they will send you the blast result. So if the file is bigger, if the sequence is bigger, you can load the file here. You click here and you look to you find here the FASTA, or probably FASTA, or at least the sequence. I don't know if they work well with GenBank, but FASTA works fine. Then let's say this is an optional, but it's good to find your result later. There is even more. You can have a big sequence, but you can limit your search to only one part, like from the 10 to the I don't know, 80 or something like that. Will not use, but you can limit to only part of the gene, of the nucleotide sequence. So this will be class 7 experiment 1. Just a name that will help us to look later. Now, next question. Which database to look? These have been changing. They periodically change these things and they organize in different ways. For example, beta coronavirus, you can guess why they put beta coronavirus now. This was not last year. Then they have this, for many people, human and mouse are the only thing that they care. If you work in medicine, probably this is the only database that you care. Human genomic and transcriptomic. You follow me with the idea, the difference between genomic and transcript database, no? Yeah, moving the head. Basically, in the genomic has introns, and the transcripts typically don't have introns. Okay, there is this RNA and intertranscribal sequence, no? Internal transcribal spacers. That is. So these are the sequences for identification for like 16S, typically 16S or 18S that we will use to identify sequences. We will going to use it in a minute. And then the standard databases that are several. Well, I think I have at least some of them. I didn't put many of them because I don't know them well and I'm not sure if they are valid anymore. But the database that okay, maybe I should first show you that was in nucleotide, but if we go to the protein side, you will have something similar, except that the sequence are, the databases are different, obviously because it's the database of protein. So let's start, kind of easier to explain from the protein. The first database that the people created is called the non-redundant protein sequence. Why is called non-redundant? Essentially because, as you remember, there are more than one site collecting sequences. So it happened, especially at the beginning when they were not coordinated, that the same protein was with one name in NCBI, with a different name in the European Bioinformatics Institute, and with a different name in the Japanese repository. And then there are other databases. There is a database called SwissProt, that is a Swiss protein database, a protein data bank, and all they are the same protein with different names. So the first thing that they did is say, okay, it doesn't make sense that we keep the same sequence with different names. Let's make a non-redundant version. So the sequence is only one time and it has in the heading all the possible names. That is what they call the non-redundant. And it's important because, I mean, people say NR to get the idea of what is NR. That is essentially not 100% correct, but essentially kind of all gene bank protein, just avoiding repetitions. Okay, if the same sequence has two names, you see the sequence only one time. Then you have reference sequence, and they started with the ref sequence. You remember RefSeq is the subset of PubMed of GenBank that is curated, that is some human took care of cleaning it. Now they have a new database that I have not explored yet, called RefSeq Select. But I understand they are doing this philosophy. Since database change with time, and at the same time you will like to, what you will like to? You would like to have the same e-value. You would like to be able to replicate the same search and get the same e-value. What they did, why is this happening here? What they did is, they created a subset of RefSeq, a selected set that they promise that they are not going to change very often. Or kind of representative protein. So RefSeq change, I mean, NR change every day. RefSeq also change often, So the e-values will change. Select, it will change less. It's a smaller subset that is kind of representative. Then they have another called landmark that they took only model organism. You know what is a model organism, no? In general, for every branch, taxonomical branch, there is kind of one organism that is kind of the representative. For example, well, human is a model organism. Mouse. Okay, mouse is kind of the model for mammals. Everything that you want to do with mammal, you don't do with dog, you don't do with elephant, you do with mouse. For fish, people use zebrafish. Everything that you want to do in fish, you can do many fish, but the zebrafish is kind of the reference. Then for plants, what is the model organism for plants?
Arabidopsis, I think.
Arabidopsis. Arabidopsis is the model of plants. Good. Then for fungus, for fungi, what is the model organism for fungi? Yeast. We will use yeast for representing all fungus. Then for bacteria, E. coli. What I am missing? I don't know. But this is the kind of animal, well, the kind of organism.
Do we have one for viruses?
I don't know. I don't know if there is a model virus. I don't know. Well, another database, the one that I say, Swissprot. Swissprot is also a manually curated, and I don't know if it's still true, but this is a team effort, of course. But the point is that the official version is the boss notebook. You know, he has the original, and he kind of curates everything manually. And it's in general considered to be a good database that people know that the protein that they are really exist. We have to say that not all proteins in R are a real protein. Many are hypothetical proteins. Sorry, there is another important, people can patent proteins. If they have an industrial application, you can have a patent of this sequence. And so that is important. If you are doing some application, you don't want to use a patented protein because you will have to pay. And in particular, if you want to patent your own sequence, you have to see that it's not patented before. Okay, let me just talk quickly about a couple one. You have the protein data bank, like Swissprot is a different database made by other people. In fact, it was who asked me? Yeah, you asked me, and it's a question that we would like to see. If you search for proteins in NCBI and you send in this order, they not necessarily give you the same result. And it will be a good exam question to compare the two databases. I don't know if it will be a homework or an exam, but I kind of want to compare sequences from two databases. Okay, what else? Environment. In general, you may know environment databases grow quickly in the last years. Essentially, when people invented the new generation sequencer and metagenomic got on fashion, people start taking samples everywhere and sequencing everywhere. So in this case, what they do is this: when you sequence DNA from environment, you can somehow, in some cases, identify proteins in these reads. But essentially, let's say that if they look very similar, if your DNA translated in the 6 reading frames, one of these reading frames look very similar to a already known protein, you can say, okay, I have not seen this protein, but I believe it's a protein. And that is what we call an hypothetical protein. It looks like a protein. We know that there is DNA that may encode for this protein, so they put in this database. And this is similar. The transcriptome, essentially, they take, I mean, this is probably an old technique, because when sequencing was expensive, instead of sequencing the genome, people sequencing the messengers. So you took all the messenger and then they assembled them and then they translated to protein. So the picture is kind of this one. At least that's what I got from their database, from their website. The NR contains all these other databases, but there are other that the metagenomic protein, the what do we say? What was the name? The environmental are outside, the transcriptome shotgun are outside, and patent. These are not included in the non-redundant. Non-redundant is big, but not necessarily containing everything. And you have smaller and bigger depending on the problem. So remember that we will like to search in the smallest database that is relevant to our case. For example, if I am looking if I am working only with a model organism, I will try to work here. If I want to work, I don't know, for some kind. In fact, I will show you the other point here, the other way to limit this one is here. Here, you have an option of limiting your search to some organism. So, for example, you as a human. And you see, I got a series of options of human gut microbiome or human gut metagenome. Let me move it a little up. But you also can see that all these objects that we find here are associated with a tax ID. And you remember tax ID are essentially numbers that identify some node, some position inside the taxonomical tree. In other words, I can also limit to, let's say, mammals. If I know the tax ID of the group of the relevant organism, I can limit and take a subset of NR limited by taxa. Not why taxonomy is important. And in my case, for example, I like to say, for example, I was looking the other time with legumes. What? No, there's another name, legumes. Or I don't know, barley. Okay, I don't know what is the name, but if necessary, you go to the taxonomy database and you look for the tax ID, and you can write the tax ID. For example, what is bacteria? Bacteria, I think is 2. Yeah, so I can write tax ID 2, and I will look only in protein that have been identified in bacteria, excluding eukarya and archaea. And of course, you see there's an option here that you can say everything except bacteria. Or what was the click button? I don't know what happened here. I click a button. I don't know what happened. I click this button, I get a new screen. Well, again,
it
going back to
says
the class.
add organism.
Yeah, but it didn't work as I expected. Well, I understand essentially that you can have more than one filter. Like you say, I want to look in mammals and fish, for example. Mammals and fish is not one taxonomical branch, but you can have more than one. Kind of telling me up.
Maybe you should write the excluding organism first. Like
no, it's something. I think it's something in the browser, or maybe I should say mammals, not excluding, and no. I don't know. I will not spend too much time trying to explain why the website doesn't work. There is other idea, exclude some, for example, exclude uncultured samples. So for me, it will make sense, given the thing that I do. It will make sense to look for non-redundant proteins that are in bacteria but not environmental, for example. You can do something like that. Bottom line is that one of the questions that you have to do, that you have to think before searching, is what is the good database to search? For two reasons. First, because you want your sequence to be there, but second, you don't want many other things to be there, so the e-values make sense. And three, maybe you want to work in something that is stable, like the landmark. The bigger databases, of course, will take more time to search. In the case of nucleotide, I show you already we have human and mouse genomic and transcriptomic, and there is the NR. There is also non-redundant, but they realized later that non-redundant was a bad name, so it's sometimes called NT. There is some ambiguity that sometimes they call NR non-redundant, but they also call NT. So that is kind of the official name in the genomic part. I went back to BLASTN and I am looking at the options in nucleotide, NRNT. Again, you have the same idea that you can limit by taxonomy, or to, well, in general, now this is even the better. You remember that we say entres. So you can put any query in this language of querying that you can use to limit your search. There is a YouTube video that you can see that they call your choosing database is not a database in the sense that it's stored, but it's a subset of the database that is relevant to your point. So you can start with all the sequences or all RefSec sequences if you like, and limit to some samples, some project to limit the way that you want to make. So instead of limited by taxonomy, you can limit for something more elaborated. Well, I show you, you can also limit to 16S sequences, and you have this RefSec for RNA and for, well, you have all this database. And well, some of these are being changing with the time. So you have to read the help. They have representative genomes and genomes in general. And the other that is interesting are this one, in particular the first one. So in many projects, people sequence the transcripts. But my point is, this database contain sequences that are essentially getting out directly from the sequencer. Okay, the ACRA database that we will see in a future class, they contain reads. Reads are the product of the sequencer. And this is the Sanger, if I'm not wrong. This is the Sanger database, the database with Sanger reads, and this is the database with the new generation sequencer. So you can even look not only in the genome, but directly in the result of the sequencing. For example, if someone did a human gut microbiota, for example, and you get all the reads, or you get a project that they have not yet finished, you can look directly in the original sequences. And of course, you have patented, and I don't know exactly how they do, but Protein Data Bank, they have a part with nucleotide. I'm not sure how they do. And the picture that they give is this one. It's much more complicated. So you have NT, and inside NT you have RefSec, and inside you have 16S, and then you have genomes that are not part of NT, and they have representative genomes, and then some classical genome patented. I the how throughput genomic things. This EST, remember, are the messengers, and these are the reads. Okay. Now, I have two examples to try. Well, we can try with the sequence that we have, but the other thing that we can try, I have some data that that is real data that produced in our department. You know Terje Steinem. He sequenced some I think 16S. I mean, you took sample and they amplify with universal primer and they get a sequence, and you will like to put a name on this. So if I am not wrong, we will see. This is a 16S. And then we have the other that I found the part of the Artemis. This one. I took, and there is an option here that you can even do, like you mark something and you have select, and what is the name? Copy. Create. No. You can save part of this as yeah, base of selection as FASTA. So I get a new window, and here is the sequence, and I can copy. Essentially, you have some option that allow you to take part of the sequence here and create a new a new file in any format. Save as FASTA or GenBank. Yeah, you will probably do something like, I will mark some region here, and I will say create a new feature with this thing. Blah. This is the feature, and then I will save this feature. No. I don't know. Save here, no? Save, no? Right. Base of selection in FASTA. Yeah, that is what I will do if I want to. You can have upstream and downstream. That will be useful later. So I can write the base of selection in FASTA format. The selection is the one that I marked here. Something like that, okay? Point is that you can, there was something else when I created apply. Okay. The point is Artemis will allow you to do something like that, that we created. This is not the one that I want. Anyway. No, you can create features here and then record them or modify them. It's not only for looking, it's also for annotating. Okay, let's go to the blast. Oh no, we finished it, no? We are out of time.
Yes, we are.
Yes, we are. Okay, there has to be homework. Well, I don't have any other thing here. I was saying, let's go to the NCBI. And I have just one comment, well, I will upload this and you can try looking for this. But what I want to propose is this. You will see that there are some options here like Megablast, discontinued Megablast, different options here, and same in Blast P, you have some alternative. Okay, so What are they? Blah, 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 blah. So homework. Explain me. The best way that you will learn this is to, now we have to somehow split them. I may split them. How can I split them? I may assign you randomly. Maybe we'll do something. I'm uploading the slides now so you can see them. But essentially we have these options in like these 5 options here and no, here, and the other 3 options here. And there is something else that you will find later called filter. There is some help here. So it's not very hard. Essentially, you have to click here and read the help, but you have to understand it and explain it with your own words. Okay? So I propose this. I will upload this now. You take a look, and I give you time to choose. If you don't choose which one you want to do, I will assign you. Okay? So you have a first chance to choose on your own, and if you don't choose, I will assign you. And only one, you don't do all of them, well, unless you want to do. But okay, this is probably one topic, and these are each one topic. In fact, there are others, but let's start with this. There are other modern blasts, but let's start with the easy ones, okay?
How much time we have? Choose.
Good question. Well, depends. I think this can be done in 1 week, but if you are very much on pressure, we can wait. I mean, it's not like I need them for the other class. My point is that you need to know them. In fact, we can even go here. You have the smart blast and primer blast and ig blast and mole blast. But okay, let's start with the simple ones. Okay. It kind of read, understand. Maybe there is a paper. Take a look at the paper and write. But maybe the basic point is that you should write it. You should write something, because later you have to write and you don't know to write. I always regret not asking a student to write more. Right? It doesn't need to be very long. It can be 1 page. It doesn't need to be, but it's important that you practice writing.
So we will
Yes or no?
choose something of the options, and we will describe what it does.
You send me an email saying the one that you choose. And if before Sunday you have not sent me an email, I will assign you something that has not been chosen.
And we will describe in writing how it works.
You read, and essentially the question, what the question here is like, why this is useful, and what is the difference with the standard blast, and maybe a little how it works. Now, the how it works can be a complicated thing, but essentially the question is like, why and how is different? No? Because if all blasts are the same, why you choose a different? So, okay. You can, in fact, for example, here we have something called primer blast. It's not very hard to guess what it's useful for, no? It's to design primers. But smart blast, I don't know the difference. What is the difference between smart blast and the normal blast? Ig blast, what is the difference?
Should we do this in the forum to see which ones are already taken?
Not necessarily. Not necessarily. Okay, if everybody chooses the same one, of course not later, but yeah, you can use the forum if you like. Yeah, why not? There is nothing secret here. Fine. Essentially, my proposal is that you read a little and write a little. Okay.